Hello, my name is Gili Osipovich. I'm a professor of dermatology and the director of the Miami Itch Center, which is the largest clinical and research center dedicated to itch. We see here patients with chronic itch of different types from all over the US and conduct clinical studies as well as molecular studies and animal models of chronic itch. The clinical studies is directed by my colleague Lena Kemper. The animal studies is directed by Duska Qiyamah, who's a world expert in animal models of chronic itch and neurophysiology of itch. And the brain imaging studies we do on chronic itch is directed by my colleague Hideki Mutoh. I would now like my colleagues to join in and describe their research in detail. Thank you.
Thank you, Dr. Yosifovich. My name is Dr. Lena Kemper, and my side of the lab focuses on our human studies of the Miami Itch Center. We do two basic types of research, the first being sponsor-initiated. This is where we work with companies to test their antipyretic treatments in several of our patient populations. These include phase 1 through 3 studies. My main part of our research lab is to do what we call investigator-initiated research. This is where we come up with our own protocols to help us better understand the mechanisms behind chronic itch. The techniques that we use include psychophysics. This is where we can induce itch and pain in our patient populations or healthy controls to better understand how these patients respond to these external stimuli. We also look at the skin of these subjects. This helps us to know how the skin is processing itch and also helps us learn where we can treat chronic itch. We do several collaborations, including with our imaging systems, and this is where we can use fMRI to actually see how the brain and central processing of chronic itch occurs. And my colleagues are going to tell you more about what they do.
Hi, this is Duska Qiyamah. I'm an assistant professor at Department of Dermatology of University of Miami. My lab uses mouse models to study itch. The advantage of using mouse model is we can easily manipulate itch pathways using strong genetic tools. Good example of genetic tool is optogenetics. You can deliver the light-sensitive channels to your specific target cells in itch pathways. Then, by shining light on your specific target cells, you can immediately activate or deactivate the target cells. So you can control itch by just turning on light. Additionally, we can monitor the neuronal cell and non-neuronal cell activity by using the electrophysiological techniques as well as calcium imaging techniques. These techniques allow us to investigate dynamic itch response at cellular levels. I have been working with Dr. Yosifovich, Dr. Mochizuki, and Dr. Lena Kemper.
Hi, my name is Hideki Mutoh. I'm assistant professor at Department of Dermatology, and I'm in charge of the Brain Research Unit at Miami Itch Center. My research unit focuses on the brain mechanism of itch in human. We conduct brain imaging research to better understand how itch-related neural signals are processed in the brain and how chronic itch affects the process. This is the MRI machine we use. This machine tells us a lot of information about the brain. For example, functional MRI is a useful method to identify which part of the brain activate while subjects perceive the itch sensation, or which part of the brain show different response between healthy subject and chronic itch patients. The other technique is structural MRI. These techniques give you a 3D or 2D picture of the anatomy of the brain. By comparing brain structure image between healthy subject and chronic patients, you can investigate how chronic itch affects the structure of the brain. I will show you how we exactly do in the MRI room. Subject lay down in the scanner like this, and then we apply itch stimuli to the subject. Then we measure brain activity while subjects perceive the itch sensation. There are many other aspects of itch phenomenon that we want to explore. In particular, we are interested in why scratching an itch is so pleasurable and why scratching can suppress itch. We believe that brain plays an important role. All these studies will eventually provide us useful information to better understand the brain mechanism of itch in humans, as well as the effect of chronic itch on the brain structure and function.
I'd like to thank my colleagues. As you can see, we do quite diverse research, spanning from human studies to animal models. This leads to collaborations with other groups in the department, as well as other departments in the medical center. For example, studies we're doing with the Bascom Palmer Eye Institute on chronic itchy eyes, studies we're doing with the Department of Psychology on itch expectations, and with our neuroimaging center. We are federally funded. We also have investigator-initiated studies that the pharma supports, as well as foundations, in particular our studies on molecular mechanisms of itch. Another area that we have focused is on educating a new generation of itch researchers, as well as clinicians interested in itch. We have quite a few researchers coming to train with us and use the models that we have developed. So it's quite an exciting time to be here, and I'm looking forward to it.